Hi everyone, this is Dr. Santosh. Today I'll be talking about concept of heterochromatin and euchromatin. Now, the concept of heterochromatin and euchromatin has been evolving. Now, I want you to tell there are two concepts of heterochromatin: a molecular concept and cytogenetic concept of heterochromatin. So, molecular concept of heterochromatin is the part of heterochromatin, part of DNA which is transcriptionally inactive. The opposite also true. The part of DNA which is transcriptionally active is called euchromatin. So what is the meaning of eu is true chromatin, true DNA, true part of DNA, true chromatin. That is the molecular concept. And hetero is other DNA. Hetero is other. So other DNA than the DNA that is transcriptionally active, you can call them as hetero. This is the molecular concept of heterochromatin. The part of chromatin that is transcriptionally active is called euchromatin. The part of DNA that is transcriptionally inactive is called heterochromatin. It can be gene containing, but if it is transcriptionally inactive, it is heterochromatin. And the heterochromatins are also last to replicate in S phase. Now, cytogenetic concept of heterochromatin is different because in the beginning of this development of this all molecular genetics, there was cytogenetic genetics. Okay, the people used to see the things through light microscope. Okay, the light microscopic view of this heterochromatic is little different from again electron microscopic view. So what is light microscopic view of euchromatin? Heterochromatin came from banding of DNA. So actually the first scientist who actually improved the banding of this DNA is his. In 1929, he saw that some part of the chromosome which is darkly stained, he called it as heterochromatin, and some part which is lightly stained, he called it as euchromatin. Now, this heterochromatin was given by his only. So euchromatin, he called that euchromatin contain gene. Okay, and this does not contain gene, no gene, heterochromatin. And now he called this chenic region, the light region, as chenic region and non-chenic region, and this euchromatin, heterochromatin. That is how today when we present chromosome like this, so we will say light band as chromosome. That you can see here. You can see the light band. This dark band is heterochromatin. It does not have gene. This band has gene. This has gene. So this is the concept of this cytogenetic concept, light microscopic concept of this heterochromatin and euchromatin. But molecular concept, even there is gene has to be transcriptionally active. That is the molecular concept of heterochromatin. Now let's start concept of heterochromatin. Let's see. The transmission electron microscopic concept of heterochromatin is packaging. When a DNA is packaged very tightly, okay, when it is tightly packed, what will you see when you throw the electron and when it is compact completely, then that will be electron dense. So electron dense region, we call it heterochromatin. So when you see transmission electron microscopic perspective, the whole chromosome is heterochromatin, irrespective of this band gene or not gene. And this concept, interphase nucleus, if you see the area of chromatin which is densely electron dense, they are called heterochromatin. So this is the concept of transmission electron microscope. Now what we conclude at last, you see, the transcriptionally this electron microscopy match very well with the interphase also and this phase also. You can see this transmission electron microscopy match very well with the molecular concept. Anything that is transcriptionally inactive, any portion of DNA that is transcriptionally inactive, the DNA has to be folded and packaged. The DNA is not, it is a very fragile type. It's a fragile type of molecule. This DNA is liable to damage. So what cell does is, the DNA, part of DNA which is not in use, they package that DNA the way we fold the threads and put in the shell. What the cell does, they package the DNA and they put that DNA generally in the periphery of this. As you see, bar bodies are also pushed into the periphery here. Bar bodies are pushed into the periphery because something we don't require, we fold and then put into the periphery. That is what is done. So this compacted, very tightly folded is heterochromatin. Because it is inactive, it is pushed towards periphery, but the center is region which is electron lucent, it is unfolded region, and this is transcriptionally active. So these are transcription, this is pushed into center. So this is electron transmission, electron microscope, goes very well with the modern concept of molecular concept. Transcriptionally active DNA will be unfolded and it will be electron lucent, and transcriptionally inactive will be folded and it will be electron dense. So this molecular concept, if you see, the whole chromosome is inactive transcription. So whole chromosome is heterochromatin in molecular point of view. But yes, if you talk in regards to presence of gene and absence of gene, so you can call this part of DNA. The light part is euchromatin and heterochromatin. But as a whole chromosome is molecular wise, it is transcriptionally inactive. At last, I'll tell you, this concept of heterochromatin and euchromatin, it is applicable only in eukaryotes, because eukaryotes only contain chromatin, not prokaryotes. And about 10% of whole human genome is permanently heterochromatin. Okay, so this is all about heterochromatin, the concepts of heterochromatin. I hope I made things more clear. Thank you.